Okay. Maybe we can start by reading the abstract first, as per the usual. Maybe it's ready, isn't it? Are there still people reading the abstract, or can we start? So for me, this is the gap sentence, but to be honest with you all, I'm not sure this is really the gap sentence, because compared to the others, I found like this one is lacking in explaining clearly what is missing in the literature. So I don't know what you think. So this is quite different from usual abstract, well, from usual gap sentence, but also the abstract actually is quite, well, normal, I would say. We are doing quite a good job at explaining, and they're also flashing some huge numbers, like millions of cells and everything, to kind of captivate the reader. So I think this is all we can say about this abstract. So basically this paper will be about presenting a method to analyze single cell data, and most importantly, this is a method that will basically, we will use this model to find which cell types are present into data we already have. So this is a good method for annotating. Right, so maybe we can start with a little bit of background and explaining what is in single cell data. I'm pretty sure you already know what is single cell, but this is my turn today to explain it to you. So let's imagine we collect a tissue. So this is a sample that comes from a body or anything else. And this tissue is basically a melting pot of different cells, right? So using a single cell data experiment, we can basically decompose this tissue into a set of different cell types. So we can say for each cell type, what are their gene expressions, so we can assess their gene profile, and most importantly, we can use this information to find similarities and differences between cells, so we can assess the heterogeneity of our cell types. So this is very useful and very valuable information. So the author wanted to use this information, and they basically wanted to download a huge set of data. So this is the flashy number we saw earlier, this million set of data. So they had some requirements for the data they wanted to use to train their tool and their model. And in this plot, basically we see that this plot represents the number of studies that were matching the criterion for their model. And as you can see here, we have this number increased over time. So today, this is estimated that there is 100 million individual cells that have been profiled using RNA-seq data. But despite this large volume of data, we are still not a model that are clearly designed to integrate cells from different data sets. So our model still lacks this ability to find similar cells across different data sets, because all of these data have been sequenced into different studies. So here the author states two requirements for a good model. So to make good use of our data, first we will need to have a model that would be able to compute a representation of cell profiles without retraining the model for each cell. So what we want is a model that we learn once, and we can use it for different cells. We can use it afterward. We don't have to retrain it every time we need to submit new data. Second requirement they state would be a metric. So what we want is a metric that can measure similarity between cells. But this metric has to be robust to noise, and it also needs to be general, so it is able to generalize across cells, so we can use it for predictions, basically. There is already methods that are available in the literature, of course, to study single-cell data. So among the unsupervised methods, we have, for example, PCA and autoencoder. So these are two famous methods. And if they are good at preserving the information in the source, so they are good for analyzing the data, one criticism that the author formulates regarding this method is their lack of ability to learn features for a cell. So they lack the ability to learn features and define what is similarities between cells. So what kind of method, well, what kind of tool will be used by the author for this publication? So they decided to use what is called a metric learning model. So this was originally used for facial recognition. So basically, what this method does is that it will compute an embedding, and in this embedding will contain the pictures of different people. What is this particularity of this embedding from? So first particularity is that the pictures of the same people will group together, but most importantly, pictures of different persons will be farther from each other. So what we found is groups clusters for one person. And nice features of this embedding is that we can use this embedding to formulate prediction. So we can feed it a new image of a person we don't know and see who it is. We can identify this person. Of course, this is for facial recognition, but later we will use it for cells, basically. So what is the model? Well, the name is similarity, or similarity, I don't know, it's kind of hard. So this is a model that will be used to perform prediction, to find similarities between cells. So how this model works is that we will have our query cells, so we will have a data set with cells we don't know. We will have also a set of references that is already included in the model. So this is also the big flashy number we saw in the abstract. This reference is composed of 23 million of cells. And what the tool will do is that basically it will find the similarity between query cells and cells that are found into the reference set. So this will give us a list of studies, say, okay, so this cell is similar to studies of the intestine, studies of in vitro data, and studies of COVID-19 lungs. And it will associate a score and also display which part of the studies seems to display a high similarity between our query. All right. So maybe we can start to dive a little bit into the model and the technical aspect of it. So we fit these cell profiles, and in this paper, they use 20,000 genes. And what the model consists of an autoencoder, so it's an encoder and a decoder neural network. So the encoder will learn features, compute a space, and the decoder will basically output this space, where we will have normally cells that are similar that we cluster together, just like objects earlier. Right. For me, the true beauty of this model is not really the decoder itself, the encoder-decoder, but more like the loss function. So this loss function will optimize two objectives at the same time. So one is a supervised triplet loss function. I will describe it later, and also an unsupervised mean square error reconstruction loss, which I will also describe, of course. So what is interesting here is that basically this whole system will combine supervised and unsupervised clustering at the same time. So we have also parameter beta that we'll describe again later. So let's start first with the triplet loss. And let's define at the beginning what is a triplet. So a triplet will be composed basically of three different cells. So one cell that we will call an anchor, and we will use two cells depending on the relationship with the anchor. So we will have a positive cell. So this is a cell that has the same cell type as our anchor, but it's coming from different study. These two cells are wrong, same cell type, right? Now we also define a negative cell. So this one is a cell that has a different cell type than our anchor, but it can come from the same or a different study. So here we compare our anchor is round, or negative cell is squared, different cell types, so they are negative. Alright, so this is the base for our model, and this is what will be used to compute the metric, the triplet. So what is the triplet loss itself? So the triplet loss will be based on the calculus of the distance. So here the distance is the squared L2 norm, and f is a function that describes the encoder. So f is our encoder. X and y represent two different set profiles. So let's call it cell X and cell Y. So basically, this triplet loss will use the distance between a cell X and a cell Y to learn similarities. So how does it lose similarities? So this is the function of the triplet loss. We see first that here we have the distance between our anchor and the positive cell, and we will compare it with the distance between our anchor and the negative cell. So let's say that at the beginning we have the distance between our anchor and the negative cells very low compared to the distance between the anchor and the positive cell. And what the triplet loss function will do is that after training, we will try to increase the distance between the anchor and the negative cells and decrease the distance between the anchor and the positive cell. But there is also a small requirement. This is this parameter alpha here. We want the distance between the anchor and the positive cell and the difference between this distance in orange and the difference between this distance in purple to be larger than this parameter alpha. So in fine, what this triplet loss function effectively does is that it will group together the cells that are similar, and it will make the cells that are different further away. So this will be the integration or this behavior of clustering that we observed earlier. Yes?
To make this triplet, we only select a single cell, one cell for each component of triplet, or select some group of cells?
Well, a triplet a triplet contains three cells, so you can see it as a group technically. Here we are, well, you have one anchor, one positive, and one negative.
Only one cell.
But one cell for each.
Oh, okay.
Yeah, yeah.
So and we randomly, so there could be several candidates to make.
Yes, there is several candidates, and there is some filtering step before. You don't like select every triplet possible, and this is actually what I'm going to explain now. But there is two kind of step of filtering step. Well, if we go back from the beginning, so we download our data from the Gene Expression Omnibus platform, right? So these data are for different tissues, different parts of the body, and they have different annotation and also different studies. We have different annotation. So annotation means a cell type, right? Because we want to do comparison across the studies, we want to avoid annotations that are present into only one study. So we will discard them. First step. And secondly, what the author will do is they will classify the ontology. So they will describe the parent-children relationship between the cell types, right? In this example, you have the T cells that are the parents of CD4, CD8, and Treg. And also CD4 is parent of Treg. But we want to avoid what they call ambiguous comparison. So when we will select our anchor or positive and negative cells, so when we will select this triplet, we want to avoid triplets that have ambiguous comparison. So what is an ambiguous comparison? It's when we compare the children with its own parents. So this is the first pre-filtering step. What we do is, for example, here we compare, in this case, the CD8 cells with T cells. CD8 are children of T cells, so we are kind of comparing 2 times the same cells, right? So we want to discard that. Same thing here. CD4 cells, we compare it with Treg being children of CD4, we discard this triplet. So we select triplets that truly define different cells. So they are chosen randomly, of course, but we still perform the filtering step to ensure that we have good comparison. So here in this case, we compare Treg with CD8 because they are truly different, right? Even though they are the same parents, T cell, but they are on the same level, if we can say that. Same here, we compare CD8 with CD4 because even if they are both children of T cells, they are different cell types. So primarily, we have the first filtering step. There is another filtering step where we want to find triplets that are also giving a lot of information for us. So in this case, the authors will classify the triplets into 3 different categories. First one would be what they call the easy negatives. So in this case, we see that the distance between the anchor cell and the positive cells is really low compared to the distance between the anchor cells and the negative cell, right? So we already satisfy the requirement of a loss function. Thus, this triplet will provide no information to the loss, and it will not train the model efficiently. There is a second type of triplet, what they call the hard negative. So this is opposite to the easy negative. In this case, the distance between the anchor cell and the negative cell is very low compared to the distance of the anchor cell and the positive cell. So this one will give a lot of information and contribute largely to the loss. But it has been found that this kind of triplets apparently contribute to model collapse. So they don't really describe what is called model collapse, but I guess this is probably some kind of situation where the model is not able to train anymore and it gets stuck at some point. Of course, we don't want that. So these triplets are rarely used in training the model. Lastly, what we will use to train our model is what they call semi-hard negative. So in that case, again, the loss function requirement is satisfied because our anchor, the distance between the anchor and the positive cell, is lower than the distance between the anchor and the negative cell. So it's satisfied. But it's barely satisfied because this distance, the difference between this distance and this distance is lower than our margin alpha that we saw earlier. So basically, this triplet will contribute a small amount of loss, but in practice, they are the ones that are often used for training the model. Is it clear for the triplet and part? Okay. So this is for the triplet loss. Now what about the second part, which is for reconstruction loss? So here we see that we use only the anchor cell, so X8, and we compare this anchor cell with the anchor cell after it has been processed by both the encoder for the function f and the decoder for the function j. So what we can imagine is that at the beginning, the difference between the original position of the cell and its position in the embedding that is computed by the model is very large. So we have a large deviation. But as we train and as we reduce the loss, this distance starts to decrease, and we have a very small deviation, meaning that the cell in our new anchor cell in our new embedding is very close to its position in the original embedding. So by doing that, we ensure that we have good reconstruction of the data. So that means that basically we are trying to minimize the destruction of biological information. We are trying to preserve variations that are present into the original data set. So we are preserving the subtle biological information, subtle differences between data sets. Is it clear for the loss function and everything? Any questions? Okay. So as I said, our loss function is parameterized using a variable called beta. So the loss function is a combination of this reconstruction loss and this triplet loss, and we can define whether we want to give more power to the triplet loss or the reconstruction loss using this parameter beta. And this is what the author wanted to determine: what is the influence of this parameter on the behavior of the model. So for that, they defined 2 kinds of tasks that the model can perform. The first one will be what they call query task. So this is where we truly assess the similarities between one cell and cells from the reference in different data sets. So in that case, we see that the score of the query task decreases as we increase the parameter beta. So lower value of beta gives a better query performance of query tasking. At the opposite, they also wanted to measure what is the influence of this parameter on what we call integration task. So in that case, we destroy the information across different samples, and we just keep the information of the cell type. So cell types we cluster together, and we don't have this clustering, we won't have clustering per studies anymore. And we see that this one behaves differently because we see that we have better integration performance as we increase the parameter beta. So if we try to visualize a little bit what I'm talking about for UMAP, so this is UMAP where here each point is a cell, and the cells are colored by the studies they are originated from, right? So when the parameter beta is equal to 0, we see that we have well-defined cluster for the studies, right? So here, for example, yellows, blue, and stuff. So this shows that this is good for query tasking, because now we can pinpoint our input cell, we can directly assimilate it to one of our data set in our reference. But if we increase the parameter gamma, we are destructuring this information, right? So now batches, I want to call it batches, but studies are mixing together. So in this case, we destroy the information of the data set, but we keep still the information of the cells, right? So we are more clustering each cells together. So this is good for integration, or for example, batch integration. So to summarize, we have two different behaviors depending on the value of the parameter beta. So when beta is equal to 0, the author says that the model acts more as a conventional autoencoder. But when the parameter beta is equal to 1, the model acts more as a batch integration tool. Right. So now we know the loss, we know how it's what is its purpose, what it does, but how do we associate a query set with a label? So basically, to do that, it's just answering a simple classification system, and for that we will use an algorithm that is called k-nearest neighbor. So in k-nearest neighbor, let's just assume that we have our cell embedding, so we have different cells that are grouping kind of together. And now we are adding a cell, but we don't know what is its cell type, right? So let's take, for example, the 10 neighbors. So in the case where we have 10 neighbors, we just assess what are the 10 cells that are the closest to our unknown cells, right? And then we will just compute what is the most abundant cell type, what is the cell type the most present into the neighboring cells. So if we count, we can see that we have 5 purple cells, we have 3 red cells, and 2 blue cells. So the cell type that is most present is the purple cells. So what that tells us is that basically we will say, okay, we have a lot of purple cells, so our unknown cells is of type purple. So this is how we decide which what is the label of our unknown cell. So if we now look at some result of the model, and we look again at the UMAP. So this time the UMAP is colored by cell type, and on the left you have the original annotation by the authors, and on the right you have the annotation that is predicted by AC similarity. Well, you see that there is a lot of similarity between these two annotations. So this is good because it shows that our red model is showing good results. There are still some differences. For example, if we look at this region with purple cells, we see that here it's more dominated with kind of green cells. So there are still some differences.
How did we get the UMAP?
How did they get the UMAP?
Yeah. From the original data set, or they used the
Sorry?
So how did they get the UMAP coordinate?
Actually, I don't think I have read about that, so I don't really know. I think they just
Because the green clusters,
Mhm.
you can see clear separation. The green two clusters, right?
Yes, indeed.
But the greens are same.
No, they are. I'm sorry, I don't think I can really answer that question. I don't know how they computed this UMAP actually. So I assume that this is. Yeah, no, I'm sorry. Maybe we can have a chat later so we can see. Right. So, well, this is so here we see that we have good similarities between the actual cell type and the predicted one. Now, what about the comparison between the other tools? So they compare the similarity a similarity with harmony, panorama, SVI, and SCRGs, which are batch integration tools. So they integrate cell types from different data sets and they try to merge them together. So to do that, they compare the tools from different data sets, so kidney, lung, and blood marrow. And they used a score that is called the average sequence score. Basically, this is a metric that measures how well cell types cluster together. Cell types cluster together. So a higher metric will mean better clustering. And we see that in this case, a similarity is performing the best among all methods. They also wanted to compare the result on two cell types, so B cells and regulatory T cells. And what they show here is that their method perform as well as harmony and SCRG, but most importantly, it perform better than Scanorama and SVI, because these two will mix cell types.
So if you use the high MML, you will get a higher ASW.
Well, normally you're supposed to get higher ASW. In that case, they fix the beta value of the model to 0.001, I think. Yeah. So they investigated how the model behaves if you vary this parameter, but they still fixed it for the generating different result. You cannot really compare if you change the parameters, right? So I assume that maybe you can have a better sequence score if you vary the parameter, you put the parameter fully to integration mode. It's also possible that they have a good sequence score here because they are still preserving a little bit of variabilities within the data set. So that means that they are not completely destroying every information, right? While harmony truly integrates stuff, so sometimes this can also lose a little bit of biological information, right? Lastly, we will talk about the case study. So there are some problems sometimes in the literature. So here the author wanted to show with a case study how well SC similarity can map a cells that we give as an input to different studies in the reference. So here they use a cell type called fibrosis associated macrophage. So this is a cell type, a cell population that has been reported in different studies of disease. But the problem is that these cells don't have a clear gene signature in the literature, so it's hard to compare between studies the presence of this cell type. So it's hard to say, okay, it's present in this study and in this study, because we don't have one metric to fit it all, right? So this is why they selected this population, and they wanted to assess the ability of their tool to find in which tissue and which condition as well their tool is able to map this population of cells. Well, they want to assess how well their tool can map this population of cells with different studies. So here we have just a bubble graph, I don't know how to call it, a bubble graph of various studies that are present into the reference. And the color represents the degrees of similarity. So the more red it is, the higher similarity we have. And what they found is that this population is highly present into disease lung samples. So this is something that has been actually observed in the literature. So this is confirming a result that has been observed in the literature. And it's also very good because we can say in which kind of lung disease this type of cells is present. So here we have, for example, COVID-19, cancer, and also some ELD lung samples. They also found this population to be present into PDAC samples. So PDAC are pancreatic tumors, basically. So with this result, we can show that we can detect a population of FM-like cells into different tissue and also different diseases as well. So this is good if we want to submit another kind of cell population. This shows that we are able to say, okay, these cells are present into this tissue or these conditions. But the authors didn't want to stop there. They also wanted to see, are we able to find, using in vivo data, to find if they are present into some in vitro experiment? It's very valuable to find in vitro in vitro experiment because we are more able to control the parameters or the parameters of the cells. We are able to control in which conditions they are cultured, and we can formulate and test hypotheses. So it's useful to identify in vitro experiment where a specific cell population is present, right? So they did that, and they were able to find some in vitro experiment where a population of cells were present. So let's recall these FM-like cells profiles were gathered from in vivo data. So that shows that they are able to bridge the in vivo in vitro gap, which is highly valuable. But also they are able to say at which day of culture we observe these cells. So in this case, we see that we have different days of cultures for in vitro cells, and we have here the FM similarity score. So how likely it is that we have some FM-like cells into this data set. And we see that this score is quite low at day 0, but it increased at day 5 and is quite high at day 8. So not only we are able to detect FM-like cells in vitro experiment, where it was in vivo at the beginning, but also we can say, okay, it appears at day 5 and we have more at day 8. So this is quite valuable information if we want to gather information about our data set. And lastly, they also used an approach called integrated gradient approach to assess what genes are important to define each cell types. So they use for that a value that they call an importance score. So what this importance score is, it tells us how much a gene greatly influences the relationship between a cell type X and a cell type Y. So supposedly, if we have a high score, that means that this gene is highly important to discriminate between cell type X and cell type Y. If we just look a little bit at this score, we see that we are only consider genes that have an expression that is higher in the cell X than in the cell Y. So we are just interested in genes unregulated in X. And well, this part is a bit more technical, but this is the part that we measure how much an increase in gene expression will participate to discriminate the cell type X from the cell type Y. So supposedly, if this value is higher, that means that these genes will greatly influence the distance between the two cells. So the two cells will be very dissimilar because similar cells are further from each other. While if we have a low score, that means they are close in the embedding. So that means that they are quite similar, but these genes doesn't really help in discriminating these cell types. So using this value, they found a set of genes, and also they could associate it with different studies. But what is interesting is that among these genes, there are some genes that are known markers of this FM-like population, and some genes that are actually novel markers. So using this score, basically, we are able to identify biomarkers of a cell type. Right. So to conclude this talk, let me summarize just some key points. First, we have a tool that is able to find similar cells that are similar from a reference set with an input set that we give it. So we are able to understand in an unknown data set what is our population, in which studies are similar, and we are able to gather some information. So we are able to gather information about the tissues and the condition as well. What is interesting with this method is that it learns similarities between cells. So it will compute an embedding that will be able to cluster together similar cells, but most importantly, we don't have to recompute this embedding every time we want to add new cells. So we can feed it with more new data, and it will generalize quite well to this data. And lastly, well, one thing that I find really interesting is that usually with machine learnings and deep learnings model, it's quite hard to analyze more than what it gives you. But in that case, we can actually go beyond that. We don't only have information about which cells are present into our data set. We also have information about which genes are important for the model to compute this embedding. So which genes are actually important to discriminate the different cell type. I think this is really valuable information. Well, this is the end of my talk. Thank you very much.
your talk. So to set the loss function, we should choose appropriate beta. So how can we choose beta? Is some kind of subjective choice, or is there any automatic way?
They didn't really describe how you can decide the beta. So I think it will be more subjective between what we want, what you want. So if you want to integrate, if you really want to integrate between batches and you don't care about the across 3D thing, you can just choose a high beta. But if you really want to gather some information about, okay, I have an unknown cell type, I don't know which cell type it is and in which conditions, so which tissues or which which IFI or disease sample it is, well, you may want to have a lower beta in that case, so you will be able to gather this
So you should
information.
enter the purpose.
Yes.
And also, I have one more simple question. So to identify the cell types, we need to set some neighborhood size like K.
Mhm.
So, and I wonder whether the result is sensitive to the choice of K or what.
So in this case, we are not falling into the KNN problems. Like this is a very simple way of classification. It's demonstrating good performance, but when you start to have like large samples, you may sometimes have you may see the limits of this algorithm. So of course, K will be very important because if you have small K, you will be highly dependent on the nearest neighbor, right? So you will be highly dependent on small errors. But if you choose a large K, yeah, you will lose in sensitivity. So that means that you will be it's less sensitive to noise, but also you might be a bit more general. You might be using some subtypes in a sense because you are losing in sensitivity, right? So I think in this method, it's like how to say. You need more to understand the KNN algorithm rather than the singularity method.
I have a question. So to estimate the distance between two different cells, you have some unified dimension. So how did they select the genes to train the model? And that is my first question. And the second question is, so each data have labels, right? They use that, but the data label was obtained by using different number of PC or different nearest neighborhood neighborhood. So they use different pre-processing pipelines. So did they care about that?
Oh, okay, so first question maybe. So the distance between cells, right? And can you just repeat the question? Distance between cells.
To train that model,
Yeah.
you have same gene set, right, from the different across the data sets, right? So with that, you need to select the unified some shared gene set from the data set. Usually, we filter out the genes which have no expression. But if you use the whole data set, you're gonna have a lot of missing values. And with that data set, they train the model. So, or did they have some, did they apply some specific algorithm to select the gene to increase the efficiency?
So actually, this is one of the criticisms I have about this paper. It's kind of hard to actually understand what they did with the data set and how they trained their model because, okay, I know this is set profiles, they use triplets and stuff, but about the true pre-processing, like what kind of normalization, is it log2 normalization, L2 normalization they used, I didn't really find this. So I think it's more in the GitHub repository where you can find the model that you will find such technical details. I know that they used only 20,000 genes, so I guess they selected them to be kind of common across studies, or at least as common as they could across studies, because they need you need a good reference set of genes to train your model, right? So yeah, but the actual processing pipeline before they feed it into the model. They didn't really describe it. To compute both the embedding, and you were talking about earlier about uniformization or uniform something. I know that this space that is computed by the encoder, so this latent phase, they want it to be uniform, unit dimension as well. So they a little bit like a PC where you know each eigenvectors is unit dimension of a space. Yeah. Well, in that case, they don't honestly they don't provide enough information to really. They don't provide a lot of information about their processing, and I think this is really one of the criticisms I have about this paper. Yeah. And the second question was the label.
Label. They use that labels, but that labels for various different various pipeline.
Yes. So in that case, again, I wasn't really satisfied with their explanation. They did say, okay, we want the labels to be present in 2 different studies because we want to compare sets across studies, so it makes sense. And also they say, okay, we want to avoid studies where the label were kind of conflictual or confusing. So they avoid studies where about cancer or about cancer, for example, for their training set. Their reference includes cells with cancer, but their training set is much smaller. I think it's only 10 million cells. Well, only. And this training set only have annotation that they selected. So I guess they did a manual curation to find good studies where they could actually trust the label. But again, they did not. It seems they don't give enough information about the processing beforehand. They don't also give that much information about how they say. Well, they do give some information about how they selected the study, but they were not caring about the ontology term. So, like what kind of disease we have, and also whether the label was a parent or a children into this hierarchical relationship I was showing you earlier.
I thought that they provided some kind of cross-validation, but they didn't, right?
No, for the processing before actually feeding the neural network, I'm kind of not happy. I feel like it would benefit from more details, or at least I didn't find them. But for the model itself, I think they did quite a lot of details. More questions, or shall we call it today?